Hello, virtual viewers, and welcome to our panel discussion. I'm Dan Ferber, Senior Editor in Scientific American's Custom Media Division. We are putting on this panel with our partner and sponsor, AstraZeneca, to explore seminal discoveries about our body's oxygen sensor system. Scientists have known for centuries that animals need oxygen to convert food into useful energy. But until recently, they did not know how cells sense and adapt to changing oxygen levels. These new discoveries have transformed our understanding of multiple diseases and have led to new strategies for treating them. We have three terrific panelists with us today. Bill Kalin is a cancer researcher who won the 2019 Nobel Prize in Physiology or Medicine with two other physician scientists for discovering how cells sense and adapt to changing oxygen availability. He is a professor of medicine at Harvard University and the Dana-Farber Cancer Institute, and he serves on the scientific advisory board of Fibrogen, which collaborates with AstraZeneca. Celeste Simon is scientific director, Abramson Family Cancer Research Institute, and Arthur H. Rubinstein, professor of cell and developmental biology at the University of Pennsylvania Perelman School of Medicine. Carrie Cooper is a nephrologist with 30 years of experience, including leadership roles at health systems and pharmaceutical companies. And he is VP, U.S. Medical Affairs, Renal Cardio at AstraZeneca. First, a bit of housekeeping. The panel discussion will run 30 minutes, and then we'll follow with a 15-minute Q&A session. To ask a speaker a question, just type it in where it says, type your questions here, and then press submit at any point during the panel, and we will answer them today. So let's get started. Celeste, why is having a hypoxia response system so essential, and how does our body respond to hypoxia?
So I think you alluded to it in your overview. Molecular oxygen is critical for many intracellular biochemical pathways, but as you mentioned, it plays an essential role in maintaining intracellular bioenergetics, what we call ATP. And so cells need to understand very quickly if they're becoming limited for this important nutrient, if you will. There are many oxygen sensing pathways, but the best characterized is the one we will be discussing in depth today. And the reason why this particular pathway is so important is that it actually coordinates thousands of pathways that help the cell, but also the tissue and the entire organism adapt to decreased levels of molecular oxygen.
Thanks, Celeste. That's really interesting. So this pathway we're talking about today, which is called the HIF pathway, and you'll hear it referred to as the HIF pathway, coordinates so many other pathways. It really plays a central role. We'll dive deeper into how it works in a bit, but it would probably be useful to rewind and talk about how scientists figured out in the first place how important this pathway was for how our body responds to low oxygen. Carrie, the first clues to the human hypoxia response came from researchers interested in studying a hormone produced by the kidney. How did this come about, and how did it launch the research that led to last year's Nobel Prize?
Yeah, thanks, Dan. And first of all, it's such a great honor to be on a panel with such esteemed scientists. As you mentioned in the beginning, I am a nephrologist, a specialist in kidney diseases, and have been caring for patients with chronic kidney disease for my entire professional career. So it's interesting even to ask the first question, why does the kidney have to do with all of this? And of course, what you're alluding to is that we now know that the hormone erythropoietin, which is primarily produced by cells in the kidney, is really the key organ ultimately governing red cell production by the bone marrow. It's a very interesting system when you think about it, about why the kidney is front and center at that. And the history of medicine tells us that we think we're smart, but clinicians and scientists have been smart for over a century. So there have been very important observations that have been noted. First of all, that if you look at the response to low levels of oxygen, so particularly it was observed over a century ago that going to altitude on tops of mountains and staying there for a period of time led to an increase in the percentage of the blood that contained red blood cells. So there was clearly some response to altitude and low levels of oxygen. It was also shown that if you made animals anemic by actually bleeding them to levels of low hemoglobin, and then took the urine from those animals, that there was something in the urine if you infused it into other animals into the bloodstream, would actually stimulate red blood cell production. And that led to the concept that there must be a hormone in the blood, which we now know to be erythropoietin, produced by the kidney, that is really the key quarterback in controlling overall red blood cell production. From the kidney perspective, of course, we know that patients with chronic kidney disease very, very commonly have anemia, low levels of hemoglobin and red blood cells. And for a long time, that has largely been thought to be due to overall deficiency of erythropoietin. But now, of course, as we understand a bit more, the concept that it isn't just that it's a sick kidney that can't make erythropoietin, but that there's a pathway for stimulating the production of erythropoietin. I think it has really changed our thinking about the pathogenesis of anemia in these patients. So, in sequence, we needed to first identify the presence of erythropoietin, which happened in the 1980s. Subsequently, the identification of hypoxia inducible factor, that is the what that actually tells erythropoietin whether to turn on or not. But now the very exciting research, I think, from Dr. Kalin and colleagues that he'll speak about is how does changes in oxygen ultimately trigger this very elegant pathway.
You began the story of the research that ultimately led to the 2019 Nobel Prize, and that was the discovery of the hypoxia-inducible factor, HIF. And Bill, you came into this story soon after that, but you came from a different angle, not the kidney angle. You came as a cancer researcher. So how did you get involved in this research on oxygen regulation?
So we began our work in this area in 1993. We knew that rare individuals who were born with a defective version of the so-called VHL gene were at high risk of developing a number of tumors, and that these tumors were notorious for producing some of the molecular distress signals that you or I would make if we suddenly weren't getting enough oxygen, such as if we went to the top of Mount Everest. And so that was a clue that the protein made with the instructions from the VHL gene had to be a critical node in the molecular circuit that allows your cells and tissues to sense whether they're getting enough oxygen or not. And that guess turned out to be correct. And so the work we were doing on understanding the VHL protein very nicely collided, if you will, in 1999 with the work my fellow laureates were doing studying the erythropoietin hormone we just spoke about. And so it was very satisfying that several groups working from different angles sort of met together in 1999. And we now knew that the VHL protein regulated this protein that Carrie just alluded to, that scientists refer to as hypoxia-inducible factor, or HIF. And then Sir Peter Ratcliffe's group and my group went on to identify the chemical signal that actually told the cell whether it was getting enough oxygen, which in turn dictated whether the VHL protein did or did not destroy the HIF protein.
Okay, thank you. So just a quick follow-up. That was great. So is it fair to say that you identified the sensing mechanism and the switch that transforms the state of the cell from one state to another?
I think that's correct. So as Carrie alluded to, we knew from the work of a number of individuals, including Dr. Semenza and Dr. Ratcliffe, as well as seminal studies done by Dr. Simon, that this HIF protein was a master regulator of various genes that you would want to turn on if you weren't getting enough oxygen. And we knew that somehow this HIF protein only accumulated under low oxygen conditions, but it was mysterious what was regulating the abundance of the HIF protein. So by the year 1999, we began to understand that the VHL protein I alluded to was sort of overseeing HIF, and that if oxygen was available, VHL would make sure that HIF was destined for destruction. Whereas when oxygen levels were low, the VHL protein would allow HIF to accumulate and now turn on those various genes, such as the erythropoietins of the world. But it was still mysterious. How did the VHL protein know, if you will, whether oxygen was or was not available, and hence whether it should or should not earmark HIF for destruction? And the work that we did and that Sir Peter Ratcliffe did showed us that when oxygen was available, a little chemical flag, which scientists refer to as prolyl hydroxylation, gets added to the HIF. And that then signals for the VHL protein to interact with HIF and mark it for destruction.
Thank you. Great explanation. So it's interesting that it's a fascinating story, and it's remarkable how sophisticated this mechanism is in the cell. And this mechanism turns on a lot of processes, a lot of biological processes. Celeste, can you take a little bit about what disease processes arise in the body when the HIF pathway malfunctions, and maybe a little bit about the biology of what happens due to this switch?
Right. So this allows me to just briefly explain how we began studying the system. So in 1995, Dr. Semenza's lab published the molecular cloning, if you will, of the two components of HIF. And it was clear that it was part of a larger superfamily, if you will, of environmental sensors that play roles in sensing oxygen levels, exposure to toxins, light-dark cycles, etc. And the way we wanted to unravel the complex biology of these environmental sensors is to start making mice that lacked each one individually. And when we saw that HIF would regulate things like erythropoietin or angiogenic factors, the things that help coordinate blood vessel development, we wondered if this was going on in the early embryo. So as an example, very frequently solid tumors develop hypoxic regions, these areas that are oxygen starved. And it was becoming clear that HIF was engaged to promote the expression of these factors that would create new blood vessels in the tumor, which would allow it to continue to grow and disseminate during metastasis. And so we wondered if maybe this was simply co-opting something that went on in embryonic development. And so we made mice that lacked the proteins, HIF in particular, and lo and behold, the embryos, these were mouse embryos, would die in utero because they couldn't make a normal blood vessel system, heart, placenta, or even normal hematopoietic cells. So this was a clue to us that it plays a critical role in central biological processes that allow the embryo to develop normally during gestation and then gets co-opted in a variety of diseases. And we would argue that low O2 is a central component of not only cancer, but other obvious examples would be heart attack, stroke, but even things like arthritis or bacterial infections or obesity. So if you think about a growing solid tumor getting to a size that can't be supported by the available bloodstream or the blood vessels, the same thing happens during obesity. The adipocytes in your fat depots begin to grow and grow and grow because they're storing way too many lipids and cholesterol and things like that. And they tax the available vascular system, creating these oxygen-starved domains as if they were a tumor. And in return, that area begins to produce factors that support inflammation. So what we're talking about is not only a key aspect of normal development and physiologic homeostasis, it underlies many, many diseases.
Terrific. Thank you. Thank you very much. That's really fascinating. It has such wide-reaching implications, and nobody even knew about it until 20, 30 years ago. That's a pretty amazing story. Bill, so your lab continues to focus on HIF pathway research in pursuit of new ways to slow cancers induced by in von Hippel-Lindau disease and I understand other cancers as well. How can this basic HIF pathway research contribute to fighting these and other cancers?
So it's been known for many decades that virtually all solid tumors eventually outgrow their blood supplies and contain regions that are profoundly low in oxygen. And so I think cancer cells, as they're expanding and growing, they have constant challenges in terms of how to get enough oxygen and how to get enough nutrients. And so I think now understanding that this oxygen sensing pathway has been, in many cases, sort of hijacked by the cancer cells is very helpful, because it allows us to begin to think about places we might intervene to sort of choke off the supply of oxygen and nutrients to the cancer cells. In addition, just knowing that the cells are hypoxic reprograms those cells and their behavior in ways that we're beginning to more fully understand. And so we and others are beginning to look at, what specific vulnerabilities are created in this very unusual hypoxic and acidic environment that most solid tumors find themselves in. And then finally, stepping back, though I think I am a cancer biologist, at least that's my day job, I think people like Dr. Simon and myself are just fascinated about the many biological effects of oxygen and, on occasion, low oxygen in normal cells and tissues.
Well, that's a great segue. I'd like to ask you a follow-up question. What are some of those effects that you like to talk about?
Well, I think for a variety of reasons, angiogenesis, the induction of new blood vessels, was one of the first places where we were able to start to think about new ways of tackling cancer. Now, of course, we were not the first to suggest that angiogenesis would be a good target in cancer. That actually goes back to the late 19th century. But it became clear that one of the ways that tumors obtain a blood supply is by turning on so-called vascular endothelial growth factor, which you can think of as sort of a fertilizer for new blood vessels. And we knew from others in the field that VEGF is under the control of this HIF protein we've been talking about today. So you can start to create in your mind a circuit that involves HIF and VEGF as being critical for the induction of new blood vessels. And fortunately, by the late 90s, there were drugs, in some cases small molecules, in some cases antibodies, being developed against VEGF. And those have proven to be very useful for a variety of solid tumors, especially kidney tumors, which are almost always linked to loss of that VHL protein we've talked about. But now we're beginning to think about targeting HIF itself. It turns out HIF is a family of proteins. Again, Dr. Simon's one of the leaders in the field here, so I'm almost embarrassed to describe this in front of her. But we know from her work and our work and others that amongst the various HIFs, although they share many common properties, they also have some distinctive properties. And we know that a particular HIF we refer to as HIF2 or HIF2 is really the bad guy, at least in kidney cancers. And there are now new drugs that target HIF2 directly that look very promising in that disease.
That's exciting. So there's some follow-on research on HIF2 that's a whole other angle. Is HIF1 and HIF2, do they overlap in their function, or do they complement each other in some way?
Yeah, so when I was a clinical doctor, we used to say there were lumpers and there were splitters. So the lumpers always look for the commonalities and the splitters always look for what's different. So I'm usually a lumper, and it is certainly true that HIF1 and HIF2 share many functions. But again, we know from the work of Celeste and others that they also have some specific functions that are unique to the different family members. Getting back to erythropoietin, erythropoietin is a very good example because we know that erythropoietin is really regulated by HIF2 and not HIF1, whereas that VEGF I talked about in most tissues, not all tissues, in most tissues is primarily under the control of HIF1. In addition, Celeste and others have taught us that there may be some special functions of the HIFs that maybe don't even have to do with the regulation of other genes, and we're just beginning to understand those.
Carrie, We've heard a lot about the function of the HIF pathway in cancer, how it goes awry, some of its basic biology. What is the significance of this hypoxia research, this curiosity-driven research in kidney care?
Yeah, thanks, Dan. For me as a nephrologist, obviously, it is really exciting to translate this groundbreaking science into what it means into actual clinical practice. The management of anemia of chronic kidney disease is really central to what we do as nephrologists, since anemia is such an important part of the lives of our patients. And I think earlier, I mentioned our approach, which obviously has been centered around replacement of the hormone erythropoietin, which we now recognize is deficient in patients with chronic kidney disease. And I think earlier I mentioned that erythropoietin can be considered, the quarterback of regulation of erythropoiesis. But I want to extend that analogy a little bit, because I think related to the HIF pathway, we now recognize that erythropoietin is not the only player in this game. And if we consider erythropoietin the quarterback, I think, as I mentioned earlier, I think it's critical to remember that a quarterback can't win the game by themselves and is surrounded by a talented group of players, and also has coaching and a head coach that oversees the entire strategy. And I think that's very relevant when we think about the HIF pathway in anemia. So clearly, erythropoietin is critical and necessary to ultimately stimulate the whole process of erythropoiesis. But let me mention iron for a second, because as you probably know, obviously red blood cells contain hemoglobin, and iron is a critical component of hemoglobin. And ultimately, normal iron metabolism is critical to producing red blood cells. Well, it turns out the HIF pathway is critically involved in iron metabolism as well. So even though we've said that HIF ultimately stimulates uh, uh, the kidney and perhaps other organs like the liver to produce erythropoietin, the HIF pathway also activates other critical pathways that ultimately improve anemia. So, in terms of iron, it's fascinating that we now realize that activation of the HIF pathway stimulates various components of iron metabolism, stimulating iron absorption from the intestine, iron mobilization from tissue stores, and various proteins that transport iron in the body. Another important parallel is another protein called hepcidin. Hepcidin is a protein produced by the liver, as you can tell by the prefix in its name. And we recognize that it is a protein that is upregulated in states of inflammation. We also know that hepcidin has negative impacts on iron metabolism. Now, why do we even mention inflammation? Well, we know in inflammatory diseases, whether it's related to infection or other rheumatologic disorders, or almost any acute illness, that hepcidin goes up. And interestingly, we know that it is very common to get worsening anemia in inflammatory diseases. Kidney disease itself, chronic kidney disease, is actually also a condition associated with upregulation of inflammatory markers and mediators. And actually, there's a substantial literature that inflammation is a cause of many comorbidities in chronic kidney disease. And hepcidin, getting back to it, is also elevated in chronic kidney disease. And we believe that hepcidin has important negative impacts on iron metabolism in patients with chronic kidney disease. So, how does that tie in with HIF? Well, it's amazing how smart the physiologic pathways are, because not only does HIF then stimulate erythropoietin, HIF increases iron metabolism, as I mentioned, and HIF also down regulates hepcidin. So you've really got a three-pronged attack. Maybe if you think about the wishbone offense in a football game, you've got three ways that you're attacking. And we like to use the term a coordinated erythropoietic response. That is, by activating the HIF pathway, you're getting all the machinery working that ultimately lets the team achieve its primary goal. In this case, not a touchdown, but actually increasing the synthesis of red blood cells to ultimately functional red blood cells with adequate hemoglobin that can deliver oxygen to tissues.
Thanks, Carrie. That was great. It really fleshes out the picture of how the hypoxia response is important in kidney disease.
So ultimately, all of this data, may help change our thinking about how we manage patients with chronic kidney disease. If I can use an analogy to another disorder, thyroid disease. If you have disease of the thyroid and your thyroid gland is not producing enough thyroid hormone, certainly, the therapy is really just to give back thyroid hormone, which is simple and very easy to do. And I would say our approach to anemia of CKD for decades has been very similar to that. We basically view it as a hormone deficiency state. We give back the deficient hormone in terms of erythropoietin and also give iron as well. But I think now we're realizing that maybe there is a more physiologic way to do it by actually taking advantage of the body's normal physiologic pathway for this and enabling the body to endogenously produce erythropoietin, which may do it in a much more sophisticated and more physiologic way. I think central to that is also the question, does the kidney still have the capacity to even produce erythropoietin? I can tell you when I trained, which was a long time ago, and we knew about anemia, and then we finally knew about chronic kidney disease associated anemia, it was really simple. Sick kidney cannot produce erythropoietin. So therefore, you must replace erythropoietin. And certainly that works. But I think, we tend to treat patients more like with a sledgehammer, where we're giving, very, very large pharmacologic doses of erythropoietin to achieve our response. Perhaps it's exciting that we might be able to now do it more elegantly and in a much more uh, physiologic way. I think one interesting example of the fact that the kidney can still do its job. So there's not all types of kidney disease are associated with the same degree of anemia. One striking example is polycystic kidney disease. And I think there are a lot of learnings from that. Polycystic kidney disease, as the name implies, is a genetic disease where the kidney over a lifetime is replaced with very large cysts, fluid-filled cysts that totally replace the normal kidney tissue. And it's interesting, they tend to compress the residual kidney tissue, impairing blood supply, and probably cause hypoxemia in parts of the kidney tissue. Well, we believe that because of that, the observation is that patients with polycystic disease often maintain their hemoglobin and red blood cell count almost normally, and we believe that perhaps that it's a demonstration that the hypoxemia in the kidney tissue can still stimulate that intrinsic renal response.
So, Carrie, I really like this idea of a more fine tuned response, replacing the sledgehammer response based on this basic research that has uncovered the intricacies of these cellular pathways. What is the advantage of doing this more fine tuned approach medically?
Dan, that's a great question, and it's something that has really been front and center to clinicians. So I think one of the problems is erythropoietin itself has many different effects in the body. It binds to receptors in many types of tissues. And even though one of its main on target roles certainly is stimulating red blood cell production, as with many hormones in the body, depending on the level of the hormone, you may start getting effects that are beyond what you want it to necessarily do. And it would take a long time to go over this, but in simple summary, we do believe that the way we currently use erythropoietin is not ideal. There are particular consequences, mostly to the cardiovascular system, that may occur with the current treatment paradigm. So I think it gets down to a simple question: is we want the right level of erythropoietin to achieve what we want to maintain hemoglobin, but not necessarily to cause some of the what we might consider off-target effects when erythropoietin is present in much higher levels than it ever is in a normal subject, when they have anemia.
Very interesting. And it points to the value, I think, of this basic research, this curiosity driven research into these molecular pathways in the cell. And maybe, who knows, it could have an effect on other tissues where doctors have to take this sort of crude approach. And in the future, with other inflammation related diseases, hopefully there'll be more fine-tuned treatments for them as well. You mentioned thyroid disease, so it's a really it seems like a really great example of the value of this basic research ultimately paying off.
Yeah, and I agree. To me, I mean, we have always heard the term bench to bedside, and this is really it's very exciting for me as a clinical scientist and somebody involved with helping to develop therapies for patients with chronic kidney disease, to actually see something that has spanned history of 100 years, but where we've seen basic science translated into research and development for patients. The fact that it can happen within not only my lifetime but within my career is extremely exciting.
If I could follow up, I think Carrie's made several very important points. One thing that's fascinated both clinicians and basic scientists alike is the fact that during adult life, the kidney is the major source of erythropoietin, but during fetal life, it's actually the liver. And then shortly after birth, the liver loses the ability to make EPO to first approximation, and the kidney takes over and is the workhorse. It was very exciting when laboratory studies started to suggest that if you could tap into this oxygen sensing pathway pharmacologically or with genetic tools you can use in the laboratory, you could reactivate erythropoietin production in the liver. And so when Carrie refers to how the knowledge of this pathway has been harnessed for clinical use, I think that's another central component of it, that now for the first time we can start to think about toggling up or down the liver's ability to make EPO to maybe pitch in when the kidney is not up to snuff.
That's really fascinating. So, to mix sports analogies, maybe a pinch hitter there.
Yes, I think listening to Carrie and Bill points up another very important aspect to all of this. HIF was molecularly cloned 25 years ago. But to be able to really capitalize on our understanding of this pathway has depended on our ability to really dissect out at a very deep level key points. For example, once again, there's more than one HIF. Even in cancer, you sometimes want to actually stimulate the pathway rather than repress it. And so I think this is a really excellent example of how deep mechanistic understanding of these pathways, which can take decades, is the only way that we're really going to be able to capitalize on our understanding for therapeutic benefits. So we see this over and over again, that simplistic ideas that come to our minds when they're first discovered don't always pay off immediately. We need to know much, much more before we can really take advantage of our understanding of the system.
Super important point. Thank you for making that point. It really is. It's the basic research so often leads to fundamental advances that are so important for humanity. So I'd like to ask one last question, and to all of you. And what is the most exciting frontier in your mind that you see opening up in hypoxia research, whether it's focused on the basic biology or disease mechanisms?
Well, I'm going to stay close to home because it's where it's still in the the management of anemia in not just chronic kidney disease patients. I would say, obviously, I think we're potentially opening the door to many types of anemic disorders. To me, the most exciting, perhaps, translation of this gets into, again, the difference between giving very large pharmacologic doses of an exogenous hormone to perhaps a much more elegant uh, physiologic approach to and as an example, I think the amount of erythropoietin that might be necessary endogenously in patients to adequately produce red cells may be very different than what we're getting when we're just using a sledgehammer approach with exogenous erythropoietin. And as with every type of therapy and pathway, it's too little, too much, or just right approach. And I'm hoping this gives us the ability to mimic what the body knows it should be doing in response to decreased oxygen carrying capacity, and correct it the same way the body would like to do.
There are so many fascinating changes in cell behavior, both normal cell and cancer cell, that are caused by changes in oxygen. And now we have sort of a richer, newer conceptual framework to start to think about, well, how is oxygen doing this mechanistically? And I'll just give you one example. Of course, the DNA in cells is not naked. It's associated with histones and then packaged into a structure we refer to as chromatin. So this HIF protein, it turns out, regulates a number of other proteins that are involved in the packaging of DNA into chromatin. And we also know that some of the enzymes in your cell that regulate chromatin themselves have some properties that strongly suggest that they might be oxygen sensitive directly. And so I think, again, oxygen is so fundamental, and now we have a new way of starting to think about how oxygen could affect the behavior of cells in biochemical detail.
That's great. And so it seems like whole new doors are opening up that could lead to who knows what in the future. So really fundamental research that needs to be followed up. And that's really fascinating. Celeste?
Right. I think there's so much more to be done. Bill alluded to other oxygen sensors. I think it's going to be essential that we understand how all these distinct oxygen sensing pathways are integrated to mount a coordinated and effective response to low O2. But there's another thing that's just become apparent to me, which is the opposite, which we have virtually no real understanding of, is what we call hyperoxia. So when in the world would that happen? Well, there's one disease state that has to do with defects in our mitochondria. These are the little powerhouses of cells that, amongst other things, generate intracellular ATP. And there's this notion that in sometimes heritable forms of mitochondrial disease, the mitochondria, the main oxygen consumer, will consume less and create a state in tissues and maybe beyond that in other parts of the organism of too much oxygen. So how does the cell and the tissue in the organism respond to that? Frankly, we have the beginnings of some understanding, but I think that's a wide open area that, frankly, I've only begun to think about myself.
That's fascinating. And too much oxygen can be toxic to some of the key molecules in a cell. Is that right?
That is correct. It can lead to severe damage. And it's the notion, amongst other things, these mitochondrial diseases probably because of the changes in oxygen levels are actually upstream of those toxic effects. So I think as we understand more about that state, that's going to be extremely important.
I want to thank all Celeste, Carrie, Bill, thank you so much for joining us today. It's been a fascinating discussion, and look forward to the Q&A, which will begin in just a minute. So here we go with the Q&A. To ask the panelists a question, just type it in the window marked Ask a Question and then press Submit. The window should be in the bottom right-hand corner of your webpage and can be opened using the sidebar menu on your left. So I'm going to sum up some of the questions. I may name some of the people who sent them in. First name only is fine. And our first question is from Susanna, and we had a couple other questions along these lines. COVID patients are very hypoxic. Is there any type of intervention that targets the HIF-1 pathway that may help these patients?
So maybe as the clinician, maybe this is Carrie, I'll take the lead on that one just from a clinical perspective. It's not surprising that we would get a question about COVID. It's obviously not a day goes by in any of our lives we don't think about COVID. And as you've pointed out, these patients are desperately ill. They have a horrible constellation of pulmonary disease, which results in impaired oxygen delivery. They often have kidney failure, some degree of acute kidney injury as part of their presentation in severely ill patients. They are clearly in probably an utmost state of inflammation when they present. So you've got probably the perfect storm for developing hypoxia, decreased red cell production. So tissue delivery of oxygen is going to be substantially impaired. And obviously, it's a little bit too new to know about whether manipulation of the HIF pathway might be of benefit in such patients. But I think it's a very reasonable question, since we know, for instance, that the ability of erythropoietin exogenously to work in these patients might be not as adequate as we would like because of the severe inflammation present. But it's a great question for future research that I'm sure will be evaluated.
And if I could just continue on that thread, I think for sure in that setting, the HIF pathway will be activated because of the bona fide hypoxia. What I don't think we're smart enough to know is whether HIF reaches a point where the HIF activation is becoming maladaptive and is contributing to the disease pathogenesis versus being adaptive. And my premise is usually, until proven otherwise, it's adaptive, because the human body is smarter than most doctors. And the reason we have this mechanism is to promote survival in a low oxygen environment. So I think that is the question, whether it's adaptive or maladaptive. But I think until proven otherwise, I assume it's adaptive. But what I can say is there's some epidemiological data that people who live at higher altitude are at lower risk of COVID. Now, there may be some confounders here, but there's even been a suggestion that HIF might indirectly regulate one of the receptors, or the key receptor for the virus. And so I'm aware of preclinical studies that are ongoing now to ask whether or not you can alter the risk of COVID in a laboratory model by affecting HIF levels.
That is very interesting. So there is a possibility down the line. So okay. So our next question semi relates to the high altitude question, because we're talking about what's happening in the blood. And the question is from Jeremy, and the question is that elite athletes sometimes blood dope with erythropoietin, EPO, to increase their stamina or performance, but anti-doping authorities now test for it. Do you foresee athletes trying to gain their performance through the HIF pathway to avoid anti-doping authorities, and what could go wrong if they do?
Dan, may I take that question?
Sure.
I think, we've discussed a better way, a more coordinated way to engage erythropoiesis. And so, the drugs we've been talking about could indeed be a better substitute. But, maybe the better strategy is what athletes have been doing for decades, which is, rather than do the pharmacologic approach, you could say, if you live in Philadelphia like me, move to Denver, Colorado, and train there for several months and have a more healthy way of doing this. As far as I know, that's still permitted. It's really trying to achieve the same goal, which is to mount a more capable oxygen-carrying bloodstream. The inhibitors that we've been talking about of the pathway that promote And in this case, it really is a HIF2 driven response in part, but also, the better iron homeostasis. It actually could be a superior approach, but it's still cheating. I think you should take a more, rather than pharmacologic approach, a physiologic approach would be my answer.
That's the key point. It is cheating. We need to athletes to play on a level playing field, of course. Thank you. Next question is from. Oh, Bill.
And I just point out that the question actually is no longer hypothetical. This has been done. A number of professional athletes have ingested research grade versions of some of these new drugs. They're buying them from offshore chemistry factories. And they've been caught in some cases. In fact, this has been written up in the New York Times a number of years ago. So the question is spot on. So some professional athletes did figure out that this might be a workaround to erythropoietin testing. And now these new prolyl hydroxylase inhibitors are being tested for as well. I think the good news is, since most of these people, you can be sure, did not have PhDs in pharmacology, and I think we're just ingesting teas or broths or whatever made from these chemicals, it's probably a pretty big therapeutic window.
That's really fascinating. What can go wrong if people try this? Obviously, you want to play on a level playing field.
For starters, if you drove your hemoglobin or hematocrit too high, you could start to get into trouble due to hyperviscosity syndrome, sludging, clotting. So that would have been at least one of the things you might have encountered if you overshot how much red blood cell mass you were creating by taking these drugs.
Thank you. So the next question, we talked about hypoxia. So as you mentioned, hypoxia in obesity, and there are other tissues this happens in. This is a question from Matteo. What about hypoxia in other tissues than the ones we talked about? Are there any diseases linked to, for example, in obesity, in amyloidosis, in fibrosis, and what other conditions?
Right. Well, the first thing that comes to my mind, because of the work we do, I mentioned in obese tissues where the adipocytes themselves begin to secrete a lot of inflammatory factors. And it's thought that this inflammation and other factors lead to a combination of inflammation and what we call fibrosis, that is, too much collagen deposition, is a key disposer for a long list of cancers. My opinion is that's why we have increasing rates of kidney cancer right now. I think we can really draw a direct link between obesity and kidney cancer. It's very clear obesity, fibrosis, cirrhosis are predisposers for liver cancer, whereas liver cancer in some parts of the world is because of hepatitis infection. I think in the Western world, liver cancer may largely be a behavioral cancer because of obesity, fibrosis, alcoholism. I think of cancer, but obesity certainly is an issue for vascular and cardiac issues. And I think some of the other panelists might want to weigh in on neural diseases, but those are the things that I think about.
Thank you. Anybody else want to add to that? If not, got a question from Cliff. The question is, to paraphrase, if increasing HIF by preventing its destruction drives tumor development, how do you increase it to treat CKD anemia? What's the difference?
Yeah. Can I try that one, Dan? So this is a question we've been thinking about for over 20 years now. So the first thing to recognize is that most drugs are drugs, meaning are useful, because you can dose them to the desired effect. There's a window, so to speak, a therapeutic window. Most drugs in common use today would not be useful if they were taken too frequently or too high of a dose. So the real question is whether there's a therapeutic window. So I think we can answer this based on empiric data and also some theoretical considerations. So when I say empirical data, for example, there are people who live chronically at high altitude or have chronic hypoxemia due to certain chronic lung conditions or heart conditions. So we can be sure those people have slightly elevated HIF for quite some time, but they're not at a conspicuously increased risk of cancer. In fact, they certainly don't look like people who, for example, have a mutation in the VHL gene. So that gives us some comfort that maybe there's a window that if you could slightly increase your red cell mass and do it safely. So that's sort of one of the empiric observations. Another was when the HIF stabilizers were being developed, much to my surprise at least, some of the compounds could turn on erythropoietin, but did not turn on VEGF, even though they're both controlled by HIF. And that was a head scratcher. But we now know that in the case of VEGF, there's another break or fail-safe mechanism that prevents HIF from turning on VEGF when it shouldn't turn on VEGF. So that was the first clue that you could start to turn on parts of the HIF program and not other parts of the program. So I think that was a very important set of observations as well. And finally, most cancers take years to decades to develop, and that's because multiple mutations have to accumulate over time, sort of like tumblers on a lock falling into place. And there's really very little evidence that HIF can act alone to cause cancer. You would presumably need many other cooperating genetic events for that to be the case. So I think there are multiple arguments for why this might be capable of being done safely.
Thank you. And it's interesting. It harks back to Carrie's earlier point about this more fine-tuned approach to therapy. There's another there's another question here from Vedad, if I'm hopefully not mispronouncing your name. And that is, do kidneys, and this is for Carrie Cooper, do kidneys sense only soluble oxygen? For example, in high altitude, low oxygen pressure, in that situation, do they sense only soluble oxygen? There's no, in high altitude, low oxygen pressure, erythropoietin, stimulates erythropoietin, but there's no anemia.
This is a great question, and I'm glad it was asked because I also wanted to kind of follow up with part of my discussion that I didn't complete fully, which was, why is the kidney at the center of all this? And if you think about it, you'd want an organ in the body that is maybe especially sensitive to hypoxemia. And for many of you, that might not seem to be the kidney. You might say, well, why isn't it muscle or something like that, where you can have periods of ischemia? But it turns out, many of you may not realize what a workhorse the kidney is, right? The kidney is working constantly. And the work the kidney does constantly is reabsorbing sodium that is filtered at the level of the glomerulus. So the work of the proximal tubule in reabsorbing sodium is a very important source of energy utilization and ATP consumption. And it turns out that especially the medullary interstitium of the kidney is one of the most hypoxic areas in the body. It's interesting. That's why people with sickle cell disease, for instance, get sickle cell crisis that can affect their kidney, and they get kidney damage because red cells really sickle within the deeper portions of the kidney where it's especially hypoxic. So it turns out the kidney is perfectly situated as a very sensitive organ. So your first part of the question is right. It really is actually the oxygen tension in these particular cells, these erythropoietin producing cells in the interstitium of the kidney, that are really where the actual sensing mechanism is taking place. So the sensing has nothing to do with anemia at all. It does have to do with the local levels of oxygen within the kidney tissue. And as Dr. Kalin said, perhaps in other organs as well that still have the ability to synthesize erythropoietin.
Thank you very much, Carrie. Well, I'm afraid that is all the questions we have time for today. I'd like to thank our panelists again, Bill Kalin, Celeste Simon, and Kerry Cooper, again, for this fascinating discussion and for their insights into our oxygen sensing system in both health and disease, and for giving us a glimpse over the horizon to the future of hypoxia research and development. It's an exciting story, and we'll be following it. Thank you to our sponsor, AstraZeneca, to our production crew, and of course, you, the audience, for joining us today. You will receive a quick survey by email to ask about your experience today. Let us know how we did. Thanks for watching, and we will see you again at our next discussion.